talking about now topic 4, organelles. Beginning with the nucleus, as it is the command center of the cell, which is right here, this sphere shaped, as you can see in the middle, the purple one. Basically, the combinations of proteins and amino acids, as it is known as the brain of the cell, as without it, really everything else can't process, as it works closely with cytoplasms and ribosomes. And then moving on to peroxisomes. They carry out oxidative reactions using cellular cell oxygen, as it works closely with the mitochondria and the endoplasmic reticulum, known as the ER. And if it fails, then cells cannot make special lipids, causing poor brain development, the loss of muscle control, in which it can also cause seizures. And then peroxisomes are going to be this little the yellow dot right there you see down the line near the end of that's our peroxisomes. Now moving on to ribosomes. Ribosomes, they are particles that carry out protein synthesis, which are these little ones down here, the yellow ones you see under the nucleus almost. They are protein synthesis that read a genetic code, as they work closely with the rough ER, folding modification, and the Golgi apparatus. If ribosomes fail to do what they're supposed to, essentially, the cell would be unable to produce essential proteins. Now, the rough ER works with ribosomes, studded with them, that synthesizes proteins. So what is that purple pass, not the dots, but the purple passageways right there, in which it synthesizes folding and the modification of proteins, as it also works closely with ribosomes and the Golgi apparatus. As in this case, if it does fail, it would be a decline in protein production and then an ability to produce cell membranes, leading to ultimate cell death, which would completely cause it to die out. Moving on to the cytoskeleton. The cytoskeleton is on the left side, as it maintains the cell shape and then assists the movement of cell parts. Up right here in the corner, we can see it, as it provides a structure, supports the cell, and also cell division, as it works closely with the ER, Golgi apparatus, as well as the mitochondria in this case. And then if the cytoskeleton does fail to produce its job, it loses a shape, no movement, also disrupts transport, and that disrupts that cell transport for cell division and cell death. Now the central vacuole, essentially they are short cylinders of microtubes, as in this case the central vacuole, it stores water, nutrients, ions, as well as waste products, as it also works closely with the ER and the Golgi apparatus. If it does fail as well, the cell will wilt down, and then it leads to like the reduced growth of storing those nutrients, water, and things like that. And then now moving on to the Golgi apparatus, as the Golgi apparatus in this case is at the bottom down here. It is this green figure, as you can see a little closer. This one right here, it processes packages modified proteins, as it also works closely with the ER and the cytoskeleton. As if the Golgi apparatus right here fails, the cells around it begin to experience impaired protein and lipid processing, leading to a breakdown in cellular transport and waste management. And then it looks like that green right here as a stack of flattened disc shape, which these ones right here, in which in this case they are somewhat coiled as well. Moving on to chloroplasts. Chloroplasts, aka known for photosynthesis, which is what we're learning right now in class. Chloroplasts They are again photosynthesis and are the process of converting light into chemical energy to create food for the plant. It works closely with the nucleus, mitochondria, and peroxisomes. If it fails, the plant essentially cannot perform photosynthesis, leading to inability to make its own food, in this case glucose, the sugar. And then it's a green oval disc-shaped double outer membrane. Moving on to vesicles. Vesicles are small membrane-bound sacs that store and transport substances. In this case, it is this guy right here. That is a vesicle, which it store and transport materials within a cell and also secrete them outside the cell. And then it also works closely with the ER and the Golgi apparatus to transport those proteins. And then if it fails to transport those proteins, the transport of molecules within and in between the cell is disrupted, leading to diseases and the overall dysfunction. It's a tiny bubble on the skin, in this case, you can see here, or a small sac-like structure inside of the cell. Moving on to the mitochondria. Mitochondria is composed of two phospholipid and protein membranes, as the structure of the mitochondria are essential for their function. And then the mitochondria is also known as like the powerhouse, almost as like the nucleus, as it is this little figure right here, this one right here. Its function is to generate most of the chemical energy that powers the cells. And then it also works with the ER, lipid droplets in plant cells and chloroplasts. As it's very important as it plays a key role in energy production, in which it also looks like this, as there's extra information on here, as you can see. That's what the mitochondria looks like, and the image right there. Now moving on to the endomembrane transport. The endomembrane system is the movement of proteins and lipids within the eukaryotic cell, involving a system of interconnected organelles such as the ER, Golgi apparatus, lyosomes, and vesicles. As essentially this process begins in the ER, where proteins and lipids are synthesized and modified, then moves to the Golgi apparatus, as we can see number 2, Golgi apparatus here, for further processing and packaging. And then finally, those vesicle buds from the Golgi apparatus carry out contacts to their final destination, such as the plasma membrane, for secretion, as we can see here. Those vesicles, number 4, those vesicles, they're brought out from the Golgi and they fuse into the plasma membrane, which process more substances and other parts for the cell. As here's also another picture for that, how the endomembrane system works, as synthesis and transport from the ER essentially are modified and sorting in the Golgi apparatus, and then packaged and transport from the Golgi containing the processed proteins and lipids, as those vesicles transport the materials or their specific cellular destinations or out of the cell.